Hello, my name is Brooks Gross. My colleague and co-organizer, Dr. Kari Ashmont, and I are program officers at the National Institute of Neurological Disorders and Stroke. We actively participate in the BRAIN Initiative and each manage portfolios of translational device grants that include BRAIN-funded projects. We are delighted to welcome you to this BRAIN Initiative Scientific Symposium titled Advances in Human Neuroscience. This session will highlight recent advances made towards understanding the human nervous system. Speakers will also present considerations they encounter in their work, ethical considerations they encounter in their work. If you have any questions for the speakers, please type them in the Q&A box during the presentations. Please address your questions to a specific speaker if possible. We will be curating your questions as they come in, and we'll do our best to answer as many questions as possible during the live Q&A session at the end of the symposium. Now, I would like to introduce our first speaker, Dr. Jen Collinger, a research biomedical engineer at the VA R&D Center of Excellence and an assistant professor in the Department of Physical Medicine and Rehabilitation at the University of Pittsburgh.
All right, well, thank you to Brooks and Kari for inviting me here to talk about some of the work that we've been doing at the University of Pittsburgh using a neuroscience-based approach to try to restore grasp function after spinal cord injury. And so now, as many of you likely know, after spinal cord injury, your level of function really depends on the level of the injury itself, and we're interested in trying to restore upper limb function to people with tetraplegia or quadriplegia who have an injury at the cervical spinal cord. And so this study that was done in 2004 demonstrated quite clearly that people with quadriplegia really prioritized restoration of arm and hand function over all other functional recovery priorities. And so our research group is trying to use brain-computer interfaces, or BCIs, to tap into brain activity that remains intact after injury to try to bridge that injured spinal cord and restore movement. And this slide here shows, sort of an overview of our approach. So we're using what we call a bidirectional BCI, where it's a circular loop that we can record activity from the brain. So shown in blue there is the motor cortex, and there's two microelectrode arrays that are implanted into motor cortex in our study. You can see an image of one off to the top right of the slide. That actually lets us record from single neurons or small groups of neurons in the brain. We can record and look at the patterns of activity and turn that into a control signal for a robotic arm. And at the same time, we can record from sensors in that robotic arm and turn that into stimulation patterns for electrode arrays that are implanted into somatosensory cortex to try to restore the sense of touch. And so we know a lot about how motor cortex is involved in controlling reaching movements and grasping movements from decades of work that had been done in non-human primates. And so, we've been doing a lot of work to try to restore reaching and simple grasping activities, and now have the ability to provide the sense of touch to somebody with spinal cord injury, where we can actually generate sensations that feel like they're coming from the person's fingers, and we can grade the intensity of those sensations. This video will give you an idea of the typical level of performance that we're able to achieve using the BCI to control this robotic arm. So the participant is in control of the endpoint velocity of the robotic arm, moving it around in three-dimensional space, as well as three-dimensional orientation of the wrist and opening and closing all of the fingers as a group. And so here they're using the BCI to perform the Action Research Arm Test. It's a test of upper limb function, where the goal of the task is to pick up the object and place it on a raised surface. And so you can see that the BCI is really enabling fairly natural and smooth control over the robotic arm. And with somatosensory feedback, this performance only improves. So using population-level recordings from motor cortex, we can actually restore simultaneous and continuous control over all of these degrees of freedom, to restore fairly natural-looking reaching and grasping movements. So what we're interested in now that we have the ability to restore some reaching movements as well as sensation, is trying to focus on control of the hand. And so we know that the ability to regulate your grasp force is critical for many activities of daily living, something adventurous like rock climbing or playing tennis, but even very simple things like holding onto your briefcase and making sure you don't drop it, or shaking hands, or, interacting with fragile objects such as cracking an egg. And so we really want to try to understand as much as we can how motor cortex is involved in controlling grasp force in order to take a biomimetic approach to restoring this function for somebody with spinal cord injury. And so today I'm going to talk about work that was done by Brian DeCleva, a postdoc in my laboratory, who really wanted to understand how grasp force was controlled across a variety of contexts. So he, within the context of our study, had the participant try to perform these different actions. So on the left, you see an image of a virtual environment where the task is to observe and attempt to control that virtual hand to grasp an object. And they were asked to grasp it with either a soft or firm force. The next iteration of that task we call the grasp in 1D Kari is that there's a cylinder that appears in the workspace either on the left or right side. The user is instructed to think about grasping it with a soft or firm force and then move it to the other target. So it's very simple. You either move left or right depending on where you start. On the right-hand side, you can see a virtual environment where the task now is to grasp in 3D Kari. And so what that means is that the targets could appear at select locations within the three-dimensional workspace. And so there was more uncertainty and complexity as to the reaching movement that would be required after the user grasped that object. And so again, this work was happening in the context of our brain-computer interface studies. So Brian was looking at neural recordings from motor cortex of a person who had a chronic spinal cord injury at the C5, C6 level. And the reason he wanted to look at this activity across a variety of contexts was because there had been some non-human primate work that really suggested that motor cortex firing was context dependent. And specifically what I'm referring to for this study referenced here, Raymond, was that the firing rates were modulated based on the range of forces that the monkeys were asked to achieve. So 2 different force levels might have a different firing rate when performed in a 2-target task as opposed to a 3-target task. So what Brian did was we had the recordings, like I mentioned. We can compute the firing rate for all of the channels across time, and then use dimensionality reduction to try to look at the overall population level activity, which is something that's commonly being done in this field right now. And so here we're just looking at the first 3 principal components of the population activity when the participant was attempting either a high force, shown in dark purple, or a low force grasp, shown in light purple. And so you can see that they have a fairly stereotyped pattern that looks very similar, except they're different in magnitude. And so what Brian did is found 2 different axes that describe 2 different types of responses that contained information about force. And so if we look at the projection of this activity onto these 2 different axes, we see what we call a transient mode. So the projection along that transient axis shows that there's a peak in activity kind of early on in the trial, right at the time of force onset. So that dashed line is showing the computer-controlled force that the user was attempting to generate. And you can see that there's a large response for the large force target in dark red and a small response for the low force target in the lighter red. And if we look at the projection onto the other axis, which we call the tonic axis, you can see that after that initial force onset, while the force is being held, that there's again a modulated activity based on the high force or low force target shown in dark or light blue. And so we consistently see these different modes of activity in our population level recordings. Now, what does it look like across the different tasks? Well, this is the data that I just showed you. I'm only going to show the high force target data right now, but again, we're looking at the magnitude of the projection of the neural activity onto this transient in red and tonic axis. If we look at this activity for the grasp plus the one-dimensional Kari, again, during that initial application of the grasp movement at the beginning there, you can see that there's a transient response in the population shown in red that looks very similar to the grasp-only task. But during the Kari phase, we do see that there's a tonic response, but it's a little bit lower in magnitude as compared to the grasp-only task. And then when we look at the 3-dimensional Kari task, again, we see a transient response initially as the grasp is applied, but during the Kari phase throughout the 3-dimensional workspace, that tonic response is reduced in magnitude. And we can quantify this by looking at the ratio of the peak magnitude in the tonic dimension as compared to the transient dimension. And what we found is that as the task complexity increased, so the number of degrees of freedom that needed to be controlled or the number of targets that were available, this ratio decreased. And so initially during the grasp-only case, the ratio was approximately 1, where the tonic and transient components had a similar magnitude, but that was decreased as the kinematic complexity was increased. And so what this meant for designing a new grasp force decoder is that we seem to have very stable and reliable information during that transient phase. So no matter what, if you were applying a high grasp force, the population response looked similar during that transient phase. But that tonic grasp force information was unreliable because it was context dependent. And so Brian came up with the idea to design a decoder that preferentially weighted this transient force information. And so he was using a standard Kalman filter to control grasp force in our BCI, where the force output is really a measure of the incoming neural activity that is transformed into force, as well as a measure of the force at the previous time step. And so the Kalman filter gain really regulates how much you weight incoming information versus historical information. And so what Brian did was insert another module in here where we can actually detect what is the probability that the user is in this transient state of actively trying to change their grasp force. And if that probability is high, we know that they want to actively change and modulate the amount of force that they're applying, so we should probably pay more attention to the incoming neural information and less so to the historical information. But conversely, if they're not in a transient state, if they're in that tonic state, we know that they probably want to just keep doing what they're doing and maintain that same grasp force. So in order to implement this, this is just one example of a task where the user was asked to pick up an object and move it back and forth across the table as many times as they could without dropping it. And using a standard Kalman filter approach, you can see that they were able to generate different forces for a high and low force target there in dark and light green, but they were dropping the object pretty consistently after 4 or 5 seconds. With the transient weighted decoder, they could maintain that performance for much longer, regularly on the order of 30 seconds. And so this is something that we're continuing to develop and quantify for a variety of tasks. But I think what Brian has shown so far is that the cortical control of grasp has both these transient and tonic components, and the tonic grasp force information is really context dependent and depends on the complexity of the task. So we're trying to use these biologically inspired decoding approaches to improve performance, and here we've shown that it can enable prolonged grasping and transport. There's a lot of work to do still to evaluate how this actually translates to functional performance on tasks. Does providing somatosensory feedback improve or change the ability to regulate grasp force? And then we want to increase the kinematic complexity of the hand, so really moving towards dexterous movements. So one last closing thought, I was asked to talk about implications for small N studies, so with a low number of participants. I think there are a lot of opportunities. We get to work with participants for many years who come in multiple times per week, and they really provide unique insights into the studies. It also allows us to really maximize the benefit and risk ratio with each participant, because we can sort of make big leaps in device development or science between participants. But of course there are challenges related to generalizability, statistical power, and trying to translate this technology for a small market. And so I'd be happy to talk about this during the Q&A session. But I'd like to thank all of the collaborators who've really contributed to this project, as well as the funding from NIH and DARPA that has allowed us to do this work. Of course, I have to thank our study participants, without whom this wouldn't be possible. So thank you for your attention. And I'd like to now just introduce our next speaker, Karim Zaghloul.
So thank you for inviting us to present our work. My name is Karim Zaghloul, and I'm an investigator in the intramural program here at the National Institutes of Health. And our lab is primarily focused on understanding the neural mechanisms that underlie human episodic memory formation. And so what I wanted to do today was describe a recent study that we just completed examining how sequences of spiking activity are replayed in the cortex when individuals retrieve memories. Now, the motivation for this work, and one of the key pieces of evidence that has shaped how we think about memory, emerges from the literature on spatial navigation in rodents. This is a schematic summary of that literature, and what we can see are the presence of what are known as place cells. These are individual neurons that respond to the locations that a rodent is in when they are traversing a spatial environment, such that when a rodent is moving along a track, what you'll see is a sequence of firing of those place cells. Now, remarkably, what happens when the mouse is asleep and at rest and perhaps consolidating their memory, or at rest and maybe thinking about the track, is that we see a replay of these sequences. And for many years, it has been inferred that these replay of spiking activity must have something to do with memory. Yet we haven't had such direct evidence that spiking replay plays an important role in human memory retrieval. Now, what we also know is that these replay events also happen in the context of this other physiological signal known as a ripple, which is a fast oscillation in the field potential recordings from the medial temporal lobes in the rodent brain. So what I'm going to show you today is a story that is comprised of 4 parts. First, I'm going to show you that we can record ripples in the human cortex and they reflect bursts of spiking activity. I'm going to show you that these spikes are ordered in a specific temporal order or sequence. I'm going to show you that when you retrieve memories, you actually replay these spike sequences, and the extent to which we see such replay is actually enhanced when we see ripples in the medial temporal lobe. The behavioral paradigm that we use for this task is what's known as a verbal paired associates memory task. So in this task, we present our subjects with pairs of words. We instruct them to form these novel associations. They solve some simple math problems as a way of distraction, and then subsequently during testing, we cue them with one word from each pair, and their job is to remember what the associated word is. And this paradigm gives us an important advantage because it gives us direct control over what memories they are trying to encode, and importantly, what memory they're trying to retrieve and when they're trying to retrieve it. And so we can see what spiking activity looks like during encoding and then during retrieval. So if we take a look at our recordings that we capture from our clinical context, we can identify these ripples. This is our 3 electrodes in the middle temporal gyrus in the cortex and an electrode in the medial temporal lobe. We've previously shown that the presence of these ripples are linked with memory formation. And now what we're going to do is we're going to ask what happens to underlying spiking activity. And so to do this, we actually implant these microelectrode arrays in our human participants who are getting clinical implants for seizure monitoring. This gives us an opportunity to record activity both at this larger spatial scale as well as at the smaller spatial scale. We primarily place these arrays in the anterior temporal lobe because this is a region that we anticipate we're going to resect in a subsequent surgery and a region that we think is involved in their seizure activity. And what we find is that every time we see a ripple event, we see a clear burst of spiking activity from multiple neurons that are recorded by this microelectrode array. And even within the array itself, if we look at the individual channels, we can see that each channel actually exhibits this small micro ripple, and the extent to which we see a ripple actually correlates with the spiking activity on that channel. Okay, so we've now seen that the ripples are associated with these bursts of spiking activity. So now let's see what happens within these bursts. So what I'm showing you here is a single example of a burst of spiking activity with the firing rate of each individual neuron shown on the right. And what we can do is we can actually reorder these neurons so we know which one fires first, and we color that blue, and which one fires last within that burst. And so now if we look at a single trial in our experiment, what we see is that every trial involves multiple bursts of spiking activity. And what we find is that the order of spiking within the first burst is actually preserved across all the subsequent bursts. So the neuron that fired first in that first burst tends to fire later in the subsequent burst. The neurons that fire later, the yellow neurons in that first burst, tend to fire later in subsequent bursts. So now we can look at what the sequence looks like in one trial. So in this trial, the subject is studying the word pair steam and seal. We could see that the blue neurons fire earlier and the yellow neurons fire later. And now we can ask what these same neurons are doing in a second different trial. And what we find is that when we observe a burst event in that second trial, the neurons have actually changed their order. So the early neurons who fired first in the first trial tend to fire later in the second trial. The neurons that fired later in that first trial tend to fire earlier in that second trial. And now we can actually quantify the extent to which sequences are similar to one another using a metric known as the matching index. We can use any metric and it gives the same result. And so what we're going to ask is how similar the sequences are for all the bursts within a given trial compared to the bursts across different trials. And what we find is that for the correct encoding trials, there's significantly more similarity within a trial than across different trials. And this is not true for the incorrect trials. So this suggests that when you're correctly encoding an item into memory, it actually involves very specific sequences of spiking activity. And so now we want to know what happens during memory retrieval. And so what I'm showing you here is an example of a trial during encoding where they are studying the word pair crow and jeep. We can see a sequence of spiking activity. As we said earlier, if we look across the entire trial, we see multiple bursts of activity that all have the same sequence. And now during retrieval, we give them the word jeep and they say the word crow. And what we find is that there are also bursts of activity that emerge during retrieval. And they have actually a very similar pattern of spiking sequence to the spiking sequences that were present during encoding. And in fact, retrieval also involves multiple bursts of spiking activity. So here, the word Jeep comes on the screen at this dashed line. They vocalize the word crow at 0. And what you can see is that there are multiple bursts of activity, all of them having very similar sequences. And as we get closer and closer to vocalization, the sequences become more and more similar to the sequences that were present during encoding. So this is true across all of our subjects and across all of our correct trials. What we found was that there was greater sequence similarity during retrieval until the time that people vocalized their response compared to the incorrect trials. The bar graph below just captures this difference between the correct and incorrect sequence similarity during this time period of retrieval. What we also find is that if we perform a simple shuffling analysis, that the sequences are specific to the individual trials that are being retrieved. If we shuffle all the trials, we find that there isn't such sequence similarity. What we also find is that during this process of successful retrieval, we actually get overall decreases in population spiking rates. And so together, what all these data suggest is that successful memory retrieval involves sparse yet very temporally precise sequences of neural activity that are replayed. Finally, we want to know how is this related to the ripples, and in particular, the ripples that we know are coupled between the cortex and the MTL. These sequences were recorded in the cortex, but in the MTL, we can actually capture individual ripples. We don't have access to the spiking activity in the MTL, but our hypothesis is that if we observe this sequence of spiking activity in the context of this cortical ripple, and if this cortical ripple happens at the same time as this MTL ripple, then perhaps those might be the times when these sequences are more temporally ordered. What I'm showing you here is another example trial. We have multiple bursts of activity. Again, from the cortical electrode, MTG, we see these multiple ripples corresponding with each of these bursts. But we also have a recording electrode in the medial temporal lobe, and we can see that there's a ripple that appears in the MTL that happens at the same time as one of the ripples in the MTG. And it turns out that this sequence here that's happening during this coupled event is actually more similar to the sequences that were present during encoding. And in fact, we find this is true across our population, that if we look at the sequences that are present during the coupled ripples, they tend to be more similar to the encoding sequences than those sequences that are present during the uncoupled ripples. So together, what these data tell us then is that this ability to replay your sequences of spiking activity is in some way associated with these ripples that are coupled between these structures. So in conclusion, what our work has shown then is that we have identified ripples in the human cortex, and they actually reflect underlying bursts of spiking activity. These spikes are ordered in a very specific temporal sequence. Memory retrieval replays this sequence, and this replay is enhanced when we observe coupled ripples in the medial temporal lobe. And so finally, I think I want to conclude by finishing up this presentation by discussing some of the ethical issues that we think are important to consider whenever engaging in this type of research. These are issues that we take quite seriously, and we spend considerable time discussing these with our colleagues both within and outside of NIH, but also with our bioethicists. There have been, of course, several good discussions about this, but I'm going to summarize 3 major points here. So first, there's a question of placing both the standard clinical electrodes as well as the special microelectrodes here for research. And I think the fundamental ethical issue comes down to balancing the additional risks associated with any electrodes versus the potential gains in fundamental knowledge, both knowledge regarding epilepsy as a disease, but also knowledge regarding human brain function. Obviously, any knowledge isn't going to be of benefit to the individual participant, but will hopefully be of benefit to future patients. In our approach, we're very careful about how we decide to place the electrodes. We only place our standard clinical electrodes in brain regions that where there's a clear indication to monitor. The additional microelectrode arrays, like the ones used in this study, involve a separate consideration, however. In our view, we believe that the risks of placing these arrays are similar to the risks of placing an additional clinical electrode, but they're not zero, which means that as best as possible, we try to place these electrodes in areas of the brain that we anticipate we're going to eventually resect. In our hands so far, what we've seen is that 80% of the time when subjects go on to receive a resection, we have indeed resected the region where these arrays were placed, and so there's no long-term consequences. However, there is a subset of patients in whom we have not performed a resection, and so we're very careful about following these patients. And so far, we've not seen any changes in seizure frequency or any other adverse events. The remaining 2 issues involve patient consent and patient autonomy. For patient consent, I think a good approach is to try to separate as much as possible between consenting the patient to the research procedure from those consenting the patient to the clinical procedure, and we try to do that. We don't want to provide the patients with any sense that there's an obligation to participate or any conflicts of interest. We have a dedicated team of research nurses that consents our participants to their research procedures, and this team is separate both from our clinical team who are discussing the surgery, but they're also separate from the actual research team that conducts the study and collects and analyzes the data. And then this finally leads to the question of patient autonomy. There is the patient autonomy, of course, at the consent stage, where it needs to be made clear that they can choose to participate or not to participate with any consequences on their clinical care. But it's also true even during testing. So we test these patients on a daily basis. However, we make it very clear to the patients that even during these testing sessions, they have the right to refuse to participate, and that should have no consequences for their clinical care. And we emphasize this to the entire clinical staff, including the nurses that are taking care of the patient, to make sure that the patient is aware that the two are completely decoupled. So these are the main issues that we consider here, and I think that we think about in terms of these patients and this type of work, but I'm happy to discuss this further if anyone's interested. And then finally, I want to conclude by thanking all of the current and former members of our lab, and in particular, I want to thank Alex Vaz, who is an outstanding PhD student in our lab, who is largely responsible for the work that I presented today. Thank you very much, and now I want to introduce our next speaker, who is Helen Mayberg. Thanks.
Thank you, Kareem. It's a pleasure to be with all of you and to present our latest work from our UH3 grant funded by the Brain Initiative. I'll be speaking about our latest findings in our examination of mechanisms of deep brain stimulation for treatment-resistant depression. Disclosures is that the basis of this work is the focus of intellectual property that's been issued to Abbott Labs. I want to start by framing the problem, because treatment-resistant depression isn't just a regular depression. We're talking in this presentation about strategies to treat patients without options. And if one's ever listened to the symptoms of a patient with this condition, one is struck by the presence of intolerable pain of the mental type and the fact that one can't seem to find the mental strategy to do much about it. And if one reaches a point of treatment resistance, it means that there are no treatment strategies that have been successful. And that's the context in which this experimental study lives. This work has been going on for many years in several of my previous labs. And it started with an idea about the relationship of what mediates negative mood in the brain and how negative mood actually hijacks intention and movement. And through a series of studies starting back 15 years ago, we implanted in a putative brain location, area 25, the subcallosal cingulate, with a hypothesis that this region was stuck on and that it mediated negative affect, and that if one blocked activity there, you could release abnormally connected cortical regions and evoke clinical improvement. And through a series of multiple iterative small experiments, we found that we could both shift acutely negative mood and affect a clinical response that evolved not immediately, but over time. And now at Emory, we have nearly a 200 patient-year experience in a small but significant cohort of patients, and have found that we can actually remediate this chronic condition, and have found that people can live without relapse for a period of many, many years. So what motivated us in these new studies was an observation that evolved as our method for implantation improved. And as we optimized the implantation procedure using tractography, we started to notice that actually one could reset the behavior very rapidly in the operating room. That if one left the stimulator off for a month after surgery, you actually had a significant carryover in the month after surgery that was only associated when you actually gave stimulation in the operating room. It wasn't induced if you did the operation asleep, and it wasn't seen without active stimulation in the operating room. The other thing that we noticed was that after a couple of months on chronic stim, that almost all patients went through a period of emotional instability, for lack of a better word, and that with minor adjustments or even no adjustments of the stimulation in some cases, it corrected. The other thing that became obvious and important to pursue was that different patients, even if at the end of 6 months were better, had variable courses. So to tune a DBS device, we basically have used the same settings for all phases of this recovery trajectory, but we see this variable rate of improvement across individuals. And so what we really need are target engagement biomarkers that can basically help us to know who to implant, how to make adjustments, to have longitudinal readouts of both the brain and the behavior, and to have that to develop rules for how to adjust the device and even think about closed-loop technologies. The work at Atlanta involved a large team, and they are one of our 3 sites. And we've implemented in the UH3 the use of prototype DBS devices that not only allow stimulation, but also sensing. We've combined measurements of local field potentials off the area of stimulation in the subcallosal cingulate with concurrent but intermittent high-density EEG to both track the biological signal of target engagement, meaning when we know when we're in the right place as a prediction, as well as how to track over time the recovery curve in patients. We combine the brain recordings with the obvious measures of self-report and the gold standard depression severity ratings, but have also started to pilot measurements, quantitative measurements of movement, of autonomic function, and of even emotional facial expression, voice, and content. What I want to just present now briefly is basically a component of what we have done and have ongoing in this study. We've now completed phase 1, which is 10 patients implanted with the PC+S system for ongoing recording of local field potentials in Area 25 over 6 months. We have ongoing experiments that will be presented in the team poster on target engagement biomarkers, both with tractography and with evoked responses off the cortical surface. I want to present briefly what we're seeing, however, in our depression state markers, where we have early clues now that there is indeed a reset that we can detect in the operating room that sets the stage for chronic changes that evolve over 6 months. This is the first presentation of our clinical outcome in this new cohort of 10 treatment-resistant patients. They're, again, a mix of men and women, middle age, onset in early adulthood, of the melancholic type, with recurrent major depression and average duration of ECT-resistant depression of about 5 years. We used our method for tractography-guided individual target localization in real time in the operating room. We confirmed in all subjects that we matched a published template that we derived from our earlier cohorts. And in this group, as you can see from the timeline of the depression recovery curve, is that 9 of the 10 patients were responders at 6 months, with 6 in remission. So having this kind of outcome really put us in a good position to track the local field potential changes over time. An important first observation was that in this study, we have been implanting both devices in the brain with the patient awake and then giving active stimulation bilaterally, really initiating therapeutic stimulation in the operating room for the purposes of tracking what exactly is that behavioral reset. We have behavioral self-reports when one is in the optimized location versus the non-optimized location on either side of the brain, but one gets an additive effect with suprathreshold stimulation bilaterally at the optimal target. And what we've shown through a machine learning approach to bands eliminated power at the local field potential measured from area 25 is that there's a multi-band change. The machine learning algorithm has extracted that what drives the change between the baseline and the end of bilateral stimulation is a beta decrease and an alpha increase on the right. So there's a bilateral beta and alpha increase on the right. It's seen in everyone, and it predicts what the carryover, the behavioral effect is over the ensuing week, with a strong correlation with the decrease in that beta signal. We have support that this may in fact be a reset, because as patients wait to start chronic stimulation for a month after insertion and testing in the operating room, we see a regression, a return of their Hamilton Depression Rating Scale score at different rates in different patients. And what we've done by now measuring off the PC+S device directly is that one can detect a return in the beta signal after about a week. And again, high beta actually is the driver of this reemergence of symptoms. Shankar has gone on to actually then characterize off the PC+S what is the driver and is beta related to the change in behavior over time that's accompanying chronic stimulation. And what we see is if one compares the relative power change in the first month of stimulation compared to the last month of stimulation, we actually see early that there's a decrease, very much like the operating room. But to our surprise, as one gets to the actual recovery measured at 6 months, it's a net beta increase. And this was quite confusing because the machine learning algorithm could not actually find a relationship between the magnitude of the behavior and the power. But what we actually came to discover was that the beta was actually switching its power and direction of its activity over time with chronic stim. So our next real focus is to understand what drives the beta switch, and that'll be the focus of our next studies. We've also tried in parallel to really look at why doesn't this beta or any of our LFP changes track with the rating itself. And what we found by exploring through video analysis what goes on in the voice, in facial expression, that actually this switch period in the LFP is accompanied by a prediction and a stability in the face and the voice that we've discerned through an analysis of behavior in the absence of patient self-report. We're now geared up to start phase 2 using the next generation RC+S platform, and we'll be continuing these kinds of studies to actually have higher fidelity data to actually track at home not only the depression severity states, but also patient self-report and ongoing fluctuations in their emotional state and in their movement with higher fidelity recordings. And I just want to end with the notion that there are some pragmatic considerations as we have these patients doing incredibly well, having participated in these very demanding experiments, but where we're using prototype devices that have no commercial support. And I think this is an important thing for us to consider, that when we actually have success in these elaborate experiments trying to understand the biology of these disorders, we now have devices that have no commercial support that are expected to be active for more than 10 years. And we need a plan that we hadn't thought of in terms of what is a patient to do that they're now tied to a prototype. What is the burden for the PIs, for the patients, for the families, for industry, for NIH, and to even consider, can an ill patient in these states really appreciate the future consequences of decisions made at the time of consent. And I think that we need as a community to be thinking about strategies of how we might even replace these prototype devices with commercial devices after these studies are done. The other thing I want to end in is that it also appears that despite these readouts, DBS is necessary, but clearly not sufficient in people who have this degree of severe psychiatric illness. And the patients themselves come to realize over time that actually they need rehabilitation. And I think we have to start thinking about how biological study design incorporates not just the engineering needs, but actually the psychotherapeutic needs. And with that, I'd like to introduce our next speaker, Chris Abbott.
I am Chris Abbott from the University of New Mexico, and I'll be summarizing our results from our brain investigation project. The title of this presentation is Neural Correlates of ECT Dosing in Depression. ECT is the most effective treatment for severe treatment-resistant depressive episodes with effect sizes around 0.8 relative to pharmacotherapy. Although transient, ECT-mediated cognitive impairment can impact all cognitive domains and delay the recovery from severe depressive episodes. Importantly, ECT-mediated cognitive impairment perpetuates the stigma with this procedure. Despite abundant ECT neuroimaging investigations to the contrary, ECT opponents cite this side effect as evidence of brain damage. The fear of cognitive impairment can be dissuasive for someone that otherwise meets the indication for this procedure. ECT clinicians individualize the dose with seizure titration, a method of limits determination of the minimum dose necessary to achieve seizure initiation. The other aspect to minimize the cognitive risk is the choice of electrode placement. Of the three commonly used electrode placements, this investigation will focus on right unilateral electrode placement, which has the least risk of neurocognitive impairment. Regarding the stimulus train, the method of limits determination necessary to initiate seizure activity is a combination of increasing the pulse train duration initially, followed by a subsequent increase in frequency to increase the number of pulses. The amplitude is held constant at either 8 or 900 milliamps. There is no clinical or scientific rationale for this fixed amplitude related to ECT. Electric field modeling has demonstrated that the 8 or 900 milliamp fixed amplitudes are 6 times greater than the amplitude sufficient for neuronal activation. Other neuromodulation treatments, such as deep brain stimulation and trans-direct current stimulation, readily recognize the importance and manipulate amplitude to achieve desired outcomes. Relevant to this investigation, amplitude also determines the electric field strength. Our study design randomized subjects to 6, 7, and 800 milliamp amplitudes with right unilateral electrode placement. Based on our preliminary data, 500 milliamp was ineffective and therefore not utilized in this design, although it is possible to use with a standard ECT device. The 800 milliamp arm was the active comparator. We assessed depression severity, cognitive performance, and imaging mid-ECT, which was defined after the 6th ECT treatment, and post-ECT, which was variable. If subjects failed to respond during their midpoint assessment, which was defined as a 25% reduction in depression severity, subjects would then receive bitemporal electrode placement for the remainder of their ECT series. The rationale for the mid-series switch to bitemporal was to ensure that the protocol would not compromise subjects' potential to respond to the ECT series. Our subject flow is summarized here. Notable findings include that the attrition rate was low, and we had equivalent attrition among the 3 arms, and the equivalent switch rate to bitemporal electrode placement. The analyses that we'll present include everybody who completed at a minimum of the 2nd visit. The 3 amplitude arms had equivalent age, sex, and clinical characteristics. This investigation did focus on an older depressed patient population characterized with unipolar depression and an age range between 50 and 80 years of age. The final charge and average seizure duration were also similar to the treatment arms. Our primary outcome measure included the Hamilton Depression Rating Scale and the percent recall from the Hopkins Verbal Learning Trials. Our antidepressant results are summarized here. We changed the pulse width with approval from our IRB, DSMB, and NIMH program officers after a planned interim analysis revealed a trend of inferior antidepressant response with the 600 milliamp arm with the ultra-brief or 0.3 millisecond pulse width. The rationale was based on the strength duration curve, which suggested the lower amplitudes may require more time for neuronal activation. Thus, we reasoned that the increased pulse width may improve the neuronal activation potential and antidepressant efficacy of the 600 milliamp arm. This reality was not materialized, unfortunately. The blue color is the 0.3 millisecond pulse width, and the green color is the 1 millisecond pulse width. And you can see in the 600 milliamp arm that the subjects really failed to respond to the increase in pulse width. The within amplitude contrasts are summarized here. The black dots are estimated marginal means, the blue bars are 95% confidence intervals, and the red arrows are for the comparison between the means. If the red comparison arrow from one mean overlaps from another group, the results are not significant. Based on the raw data noted above and the within amplitude contrast, the red box highlights the 600 milliamp arm, which had initial improvement, but unfortunate plateau between the mid and post-ECT assessments. The between amplitude contrasts are summarized here. The second red box on your far right highlights the post-ECT Hamilton Depression Rating Scale differences that demonstrate that the 600 milliamp arm had inferior antidepressant outcomes relative to both the 7 and 800 milliamp arms. Our primary cognitive measure, which again was the percent recall from the Hopkins Verbal Learning Trials, did not demonstrate cognitive impairment, and this was consistent with other measures of the Hopkins Verbal Learning Trial. You can appreciate on the red lines amongst the different amplitudes how the results really did not demonstrate any cognitive impairment throughout the ECT series with right unilateral electrode placement. Our secondary cognitive measures did demonstrate amplitude-mediated cognitive impairment with the 800 milliamp arms. This was best demonstrated with phonemic fluency, which is illustrated here. These results were also similar with the category fluency from the same neuropsychological measure. The red box illustrates the cognitive impairment notable within the 800 milliamp arm. They had a dramatic reduction in phonemic fluency performance relative to the pre-ECT to mid-ECT measure, and this impairment persisted to the end of the ECT treatment. In addition to clinical outcomes, the goal of this investigation was to understand the relationships between pulse amplitude, hippocampal electric field strength, hippocampal neuroplasticity, and clinical outcomes. The importance of delineating these relationships is illustrated with this slide. Hippocampal electric field strength is variable despite constant current delivered outside the skull. The red box illustrates the degree of overlap with right hippocampal electric field strength between our 6, 7, and 800 milliamp arms. We assessed the relationship with electric field, neuroplasticity, and clinical outcomes with the global ECT MRI collaboration database with 190 subjects that completed ECT with a right unilateral electrode placement. The results demonstrated a linear relationship with the weaker left hippocampal electric fields, which is contralateral to the right hemisphere stimulus delivery and potentially was related to a ceiling effect on the right hippocampus. We did not find a relationship with antidepressant outcomes, which may be related to a selection bias related to the Gemrick database. The selection bias is related to high response rates for some of the sites, which can be up to 100%, and not reflective of routine clinical ECT care. We replicated this relationship between electric field strength and neuroplasticity with our Brain Initiative project. We included the electric field from the final post-ECT assessment, which may include bitemporal electrode placement, as time and treatment number are critical variables when assessing changes in neuroplasticity. In contrast to the Gemrick sample, which included higher pulse amplitudes between 8 and 900 milliamps, and a linear relationship with the electric field and neuroplasticity in the left hippocampus, our sample included pulse amplitudes that were much lower, between 6 and 800 milliamps, and demonstrated an equivalent linear relationship between electric field and neuroplasticity in the right hippocampus. Electric field strength and antidepressant outcomes did not demonstrate a linear relationship. However, extending our GEMRIC analysis, the changes in right hippocampal neuroplasticity and antidepressant outcome demonstrated a very strong linear relationship. Going back to our cognitive outcomes, the electric field strength was related to cognitive outcomes with the phonemic fluency measure. The relationship between changes in right hippocampal neuroplasticity and cognitive outcomes depicted on your right was a similar direction but less robust. In summary, our results suggest a trade-off between antidepressant outcomes, where higher amplitudes may be better, and cognitive outcomes, where lower amplitudes are better. Further, the imprecise variable electric fields are associated with fixed amplitudes. The increased electric field strength associated with hippocampal neuroplasticity, and the hippocampal neuroplasticity is related to antidepressant outcomes. The sweet spot of hippocampal neuroplasticity for optimal clinical outcomes may be achieved with precise ECT dosing. For our next step, we'll be using amplitude titration to individualize this precise ECT dose and reduce the risk of cognitive impairment while maintaining the antidepressant outcomes. In conclusion, I'd like to acknowledge our grant support with our project ECT Current Amplitude and Medial Temporal Lobe Engagement, as well as our collaborators, Dr. Davin Quinn at UNM, Dr. Eric Gerhardt at UNM, Dr. Z.D. Dang at NIMH, Dr. Sean McClintock at UT Southwestern, Dr. Mikuláš Arglin at Zucker Hillside. Also our UNM Psychiatric Research Group and our UNM ECT service, and most importantly, gratitude to our ECT participants. And now I'd like to introduce our next speaker, Dr. Ted Satterwhite.
Thank you. So today I'll be talking about individual variation of association network topography in youth. So I'm a psychiatrist, and my lab primarily studies the developing brain. The reason we do this is that the early onset of most psychiatric disorders and multiple sources of data from animal studies, epidemiologic studies, and translational studies in humans suggest that psychiatric disorders can be conceptualized as disorders of brain development. So the overall program of research we go for is to understand how the brain develops normally and how abnormal brain development is associated with different forms of psychopathology. The goal here is to describe mental illness in terms of these abnormal trajectories of brain development so we can intervene earlier and get better outcomes for our patients. Now, clearly, all of this research has important ethical considerations because we're trying to develop predictive biomarkers in children. Today, I'm going to be talking about recent work that focuses on large-scale functional brain networks. This has become one of the dominant lenses for understanding human brain organization. Work using functional MRI in humans has shown that large-scale functional brain networks at the macro scale are highly reproducible. So here you can see work from Thomas Yeo and Randy Buckner that shows that large-scale functional brain networks are extremely reproducible at the group level. So these functional MRI can pull out perceptual systems like the visual system, the motor system, as you can see there in purple and in blue. However, it can also pull out higher order functional networks that are responsible for cognitive functions like executive function, which is critical in the developing brain. However, typically when we look at these large-scale functional networks, as a field, we use them in a fairly one-size-fits-all sort of way. And this assumes that each functional network is in the same location on the anatomic cortex of each individual. However, recent work in adults from multiple independent laboratories has shown that this assumption is demonstrably false. So work from different laboratories using different data sets and different methods has shown that the spatial layout, which is called the functional topography of these large-scale functional networks, varies substantially across individuals. So where these functional networks are located, their functional topography varies between individuals. Furthermore, multiple convergent lines of evidence has shown that this variation itself is heterogeneous, and different parts of the cortex have different amount of variability. In particular, higher order networks responsible for executive function and complex cognition are the most variable. So you can see here in yellow on the left, again, the frontoparietal network, which is responsible for executive function. This tends to be one of the most variable networks, and this has been seen in multiple independent adult data sets. However, it wasn't really known how this functional topography, this individual variation in functional network layout, evolves in development and associates with individual differences that we care about, like executive function. And one of the reasons we care about executive function and other higher order cognitive capabilities is that they're linked to all sorts of outcomes that we care about for kids. So executive function is a broad cognitive domain. It includes things like impulse control, cognitive control, working memory, and is linked to educational attainment, initiation of drug use, violence, et cetera. And so if we can understand how functional topography develops in kids and is linked to these outcomes, it could be an important advance. So this is what my postdoc, Zaishu Kui, tackled in this project. And to address this question, he used data from the Philadelphia Neurodevelopmental Cohort. The Philadelphia Neurodevelopmental Cohort, or PNC, this is the study, not the bank. It spans ages 8 to 22. It includes an even mix of males and females, and an even mix of Caucasians and African Americans. It was led at Penn by Raquel Gurr. And to delineate individualized functional networks, we used a recently developed machine learning technique called non-negative matrix factorization with spatial regularization. This was developed by our collaborators, Hongming Lee and Yong Fan, and in a previous work, they've shown that using resting state data from the Human Connectome project, they can pull out individual specific networks that align extremely well with individual patterns of task fMRI activation. So here, in a sample of nearly 700 kids from the Philadelphia Neurodevelopmental Cohort, we use this non-negative matrix factorization to define individualized networks. And the first step of this is to take the 27 minutes of very high-quality fMRI data we have and build a group atlas. And the reason for this is to ensure that the networks are present in all individuals and have correspondence across individuals so we can analyze the data. And the group atlas that we pull out is very similar to other group atlases that have been published in the literature. However, the key thing, the next step, is that then that group atlas is tailored to each individual's functional neuroanatomy. And when you look at individual participant data, you can see that there actually is a tremendous amount of variation in this functional topography. So here on the left, we see on this slide, we see the group atlas. And what's important to know is that this non-negative matrix factorization technique produces a soft parcellation, so each vertex gets a loading for each network. This can be discretized for display, but really it actually has more power as a soft parcellation. And if you look at how these networks are distributed in individuals, you can see in these higher order networks responsible for executive function, like the frontoparietal network and the ventral attention network on that bottom row, that there's a tremendous amount of spatial variability in how they're laid out in individuals. Now, we can look at this statistically across the entire sample of nearly 700 kids, and we can see that just as in studies of adults, the network variability is maximal in association cortex. So you can see here on the left-hand panel that the variability is maximum in frontoparietal cortex and other higher order association cortex, and then you can quantify this network by network across the 17 networks that we defined on the right-hand panel. And again, the frontoparietal network is the most variable, but also other association networks are much more variable than low-order networks like the visual system or the motor system on the left hand of that violin plot. So we've basically replicated findings in adults so far and shown that there's a lot of variability in the functional topography, and the variability is maximal in higher order association cortex, especially that's the parts of the networks that are responsible for executive function. So next, though, what we want to understand is how this functional topography related to things we cared about, like executive function, and changed with age. So one first crack at the data we took was to summarize this very complex data in a very simple way. So we derived a summary measure that we call the total network representation. The total network representation is literally the sum of the weights of each functional network over the entire cortex. It basically summarizes how much real estate each individualized network takes up on a given person's cortex. Now, one important point is this is all done in normalized space, so we're not aliasing in individual differences in cortical surface area. So this basically says, okay, this total network representation says how much a given network is taking up on the cortex of a given individual. And what we found was that this total network representation doesn't change much over the age range that we study, between 8 and 22. It's not like these networks are expanding or contracting dramatically over the adolescent period. However, we did find that total network representation was associated with executive function while controlling for age. So in fact, what we found is that the networks that were responsible for executive function, like here on the left-hand panel, the two of the frontoparietal networks, individuals who have more total network representation of those executive networks perform better on executive tasks. So if they have more real estate devoted to the executive networks, they're performing better at executive function. So this is a very, very simple measure, as I said. Next, we wanted to look at the overall complex multivariate pattern of functional topography. And we found, looking at the overall high-dimensional data, that we could predict executive function with a reasonable degree of accuracy in completely unseen data. To do this, we use split half ridge regression with nested parameter tuning. And what we found is that we can predict executive function in the second half of the data the model hadn't seen, and vice versa. So we see prediction correlations of 0.46 in complete out-of-sample testing. This is well beyond the precision of our permutation testing procedure. And we were actually pretty pleased with this. This is a higher level prediction that we often see from functional MRI, and shows that this pattern of functional topography has important implications for behavior. However, as a next step, we wanted to see whether again, this pattern of functional topography was related to age. And previously, when we were looking at the total network representation, we saw no relationship. And so somewhat to our surprise, we saw that actually we could predict an individual child's age with an even higher degree of accuracy than we could predict their executive function, again in complete out-of-sample testing. Here, we could predict an individual's child's age within a 2-year margin of error in a correlation of around 0.66 in out-of-sample testing. So that was somewhat surprising to us because we know from the total network representation that these networks aren't expanding or contracting much over development. So what could be driving this effect? So Zeshu did a number of analyses to try to investigate this. And what he found was that the features that were driving the model to be able to decode an unseen individual's age were present in association cortex. And the same association cortex, in fact, that is most variable across individuals. So the association cortex topography that varies the most among kids also is what allows us to predict their age. We dug a little further, and all the details are in the paper. And what seems to be going on here is that association network topography at the edges of the network is being refined over development, even though the gross amount of area that they're taking up isn't expanding or contracting. So as a final step, we wanted to understand how this network variability, which is related to age and predicts executive function, is constrained by fundamental properties of brain organization. So here, what we did is we tried to understand the relationship between network variability and fundamental properties like evolutionary expansion, cortical myelin content, and cerebral blood flow. And to do this, we used an extremely conservative spatial permutation testing procedure developed by Aaron Alexander Block. And we plotted the network variability here on the y-axis of each of these plots versus evolutionary expansion, versus cortical myelin content, and versus mean cerebral blood flow. And what you can see is that these highly variable networks also have a high level of evolutionary expansion, have a low cortical myelin content, and elevated cerebral blood flow. So I'm going to summarize these results and try to put them together for you. So first, we found that in adults, functional network topography is highest in association networks in youth. That the spatial layout of these functional networks varies a lot across individuals, just like we see in adults, and these networks tend to be most variable in higher order association cortex. When we look across the age span studied, we found that kids who were better at executive functioning had greater total network representation of the networks that are devoted to executive function, like the frontoparietal network. Furthermore, we found that this complex functional topography of association networks allowed us to predict the age of individuals and the executive performance of individuals in unseen data using ridge regression in unseen data. Finally, the cortex that allowed us to predict age and executive function that is also the most variable in individuals, we found is the same cortex that is expanded through evolution, is lightly myelinated, and receives disproportionate cerebral blood flow. If you put all this together, the results are remarkably consistent with a model developed by Fenna Kreenan and Randy Buckner, whereby in evolution, association networks become untethered from rigid hardwired developmental programming. And the thought is that this loss of tethering and lack of hardwiring allows for greater adaptability in development, but also comes at higher metabolic costs and also potential vulnerability to neuropsychiatric syndromes. So this work has motivated a lot of stuff we're working on now, as well as things we're planning. This includes relating it directly to psychopathology, going from cognitive function to clinical symptoms. And so currently, Zaishu is working on a paper where he uses multi-view machine learning techniques to link this diversity of functional topography to different measures of psychopathology in a data-driven way. We're currently working with Mike Millam in the Healthy Brain Network data to try to replicate and generalize these results. And we're planning to look at how these individual-specific functional networks evolve on a person-specific basis over time using data from the Adolescent Brain and Cognitive Development Project in collaboration with Damien Fair. Finally, and perhaps most excitingly, in work funded by the Brain Initiative, our collaborators Desmond Oates and Yong Fan are using these individual-specific networks to target TMS. So I want to end there by thanking the huge team that is required for this sort of work, in particular Zaishu Kui, the postdoc who did literally all of the work for this project. I want to thank Raquel Gurr, the overall PI of the PNC, and our collaborators in radiology and bioengineering, Yong Fan and Hongming Lee, who developed the non-negative matrix factorization technique. Lastly, I want to thank our funding sources, in particular the Brain Initiative and NIMH, who's made all of this research possible, as well as our study participants and their families. Thank you.
Great. Thank you all so much for those wonderful talks. And I want to extend a thank you also to the audience for your patience with us as we work through some of these technical challenges today. We're going to open it now for some live question and answers. I'm going to start with Chris. So Chris, for your ECT amplitude dosing, you looked at sort of the trade-off between clinical outcomes and side effects. And I'm wondering if you can talk a little bit about what you think might be a specific amplitude for optimal clinical outcomes based on the work that you've done and the results that you've seen so far.
Sure, thank you. Looking at the trade-off between the 6, 7, and 800 milliamp arms, when we did an older patient population between 50 and 80 years of age, it looks like 700 represents a cutoff where the inflection point about non-response shifts to increased ECT response. This was also consistent with the cognitive side effects looking at phonemic as well as category fluency, where the 700 milliamp did seem to have less cognitive impairment. However, there is tremendous variability. Some of the 700 milliamp subjects had an E field specifically in the hippocampus that mirrored the 600 milliamp arms. So prior to recommending a different amplitude as routine clinical care, I think we need to take a step back and really try and develop a method for improving the precision of ECT dosing. Basically, what happens outside the skull is constant, but inside the skull is highly variable. And we really want to flip that, maybe have individualized amplitudes to achieve the same endpoint about what's happening inside the skull. Methods to do that would be E-field informed ECT, which would have limited translational impact. We would really like to employ some of the work of Dr. Lisenby with amplitude titration and come up with a multiplier to where we can do amplitude titration to standardize the dose and then incorporate a neuroplasticity multiplier.
Thank you, Chris. The next question is for Helen Mayberg. Do you think that there is a change in receptibility to rehabilitation following the DBS treatment, and have any of the patients seen changes in their psychiatric treatment efficacy pre or post DBS?
Thank you, Brooks. That's an interesting question and kind of resonates with what we observe. These people, by definition, have failed ECT, psychotherapy, and at least 4 medications, and it's remarkable the absence almost of side effects to really extreme doses of medications that they're on, prior to entering the study, they can attempt to do psychotherapy, but it's just ineffective. As they're not demented, there's a disconnect between the thought and the training and actually have it hold. And what we notice is that even from the beginning, they have capacity to take on more activities. So interventions like behavioral activation can be really the first course because they really don't yet seem to have the machinery to engage in the kind of rehabilitation strategies that the full-blown cognitive behavioral therapy requires. But actually, after 6-8 weeks, it seems to shift. And we're really interested to know if our changes in the brain going from a low beta to a high beta might reflect this transition. So yes, they become more responsive even years out. They don't tend to relapse. And when there's minor fluctuations, one can make dose adjustments in their medication. Actually, we never change the dose of the DBS over years once we get an effective dose. So the answer is yes.
Thank you, Helen. Our next question is coming in for Kareem. So Kareem, since you are studying patients with epilepsy, do you make or see any distinction between the physiological and pathological ripples that you may actually be recording from the intracranial EEG?
So thank you for the question. I think it's a very good question. As you noted, we are recording from patients who do have epilepsy. And so in those patients, we do often see pathological activity, both intraictal discharges as well as pathological ripples. People have studied these ripples, these pathological ripples, and there has been some suggestion in the literature that they actually are detrimental rather than beneficial for cognition. We, in our work, generally, when we are thinking about normal memory retrieval, we try as best as possible to remove pathological events from our data analysis. So we try to exclude electrodes that are involved in pathological activity, as well as individual trials that exhibit. What we find is in the ripples that we capture, we see these sequences of spiking activity that are actually relevant for memory retrieval. We hypothesize, but we haven't actually shown this yet, but we plan to do this. We hypothesize that if we were to look at the pathological ripples, what we'd expect to see are sequences of spiking activity that are actually not organized in a consistent manner, or that may not be organized in a way that is beneficial for memory. Again, we haven't really done this, but that would be sort of our working hypothesis at this point.
Thanks, Kareem. The next question is for Ted. Do individual differences in functional topography relate in any way to underlying anatomical or structural differences across individuals, or are they distinct?
Yeah, that's an excellent question. I think one thing to note at the beginning is that there is a lot of anatomic variability in the same places that there's differences in functional topography. However, the tools that we've used for a while are much better at handling them. So modern image registration techniques with deformable registrations and surface-based registrations can line up the structural brains quite well. And so techniques have accounted for that, and we've been able to study that in the past. And previously, we've just ignored the functional variability and assumed that by lining up brain structure, we're also lining up brain function. And so this is a different axis of variability, the structural variability that has been previously ignored. And by not accounting for this, we're basically aliasing in the spatial distribution of our variability in functional networks to any measurement of connectivity. So I think they're both important sources of variability. It's just one hadn't been well studied in the past.
Great. And our last question for Jen. Jen, do the transient and tonic components represent motor intent in the data that you are collecting, or do you think that they could be related to the brain processing of sensory events?
Yeah, thanks. That's a really interesting question, and I'm not sure that we can really ever completely separate motor intent from sensory feedback because those are so tightly linked together. I think the evidence that we have suggests that these are primarily representing motor intent, and I can tell you a few reasons why. One is we are able to record from some areas in somatosensory cortex, and while we do see that there is a transient and tonic response, it tends to be more muted than what we're measuring in motor cortex, at least for this particular task. But if you remember that, we're working with people with spinal cord injury who are really imagining and attempting to perform these movements. So they're not getting any actual sensory feedback during this task. And so what we're measuring here is really, kind of that forward model command to the limb, I think. It's hard to say because these participants do have traumatic spinal cord injury and in the past have actually grasped with their own hands. And so, it may be that they have this efferent communication or afferent communication going back and forth between somatosensory and motor cortex that has driven these memories. But right now we're seeing these responses in the absence of any actual somatosensory feedback.
Thank you, Jen. The next question I have is for Helen. Did you find anything in your data so far that you did not expect?
Well, I think you always find things in your data you don't expect, but I think that what has been helpful for us is to actually let the data tell us what we should see rather than actually thinking we knew what we were going to see. And this switch between a signal that happened early actually transitioned and actually morphed into a different kind of signal late, and the appreciation that the recovery wasn't linear, neither at the clinical level, but neither at the brain level. And I think it really gives us pause about how to think about what these devices do. Do they reset the brain and the rest is all rehab? Are we looking at acute changes and then plasticity changes? I think we have to have different models, and that's really something we have to pay more attention to now.
Great. This question is for Karim again. Which microelectrode, array did you use, and where do you think these units are located within the brain?
So the microelectrode array that we used was the what's colloquially known as the Utah microelectrode array. It's an array of 96 microelectrodes arranged in a 4 by 4 millimeter grid. So each microelectrode is spaced from its neighbor by approximately 400 microns. We place these electrodes in the vast majority of cases in the middle temporal gyrus of the anterior temporal lobe, so the lateral temporal cortex, just because that is the area that we have access to when we're performing our initial surgery for placement of the clinical contacts. Again, largely we are placing these microelectrodes in this region because this is an area that we anticipate that we're going to resect, and so as best as possible we try to put these arrays in a region of the brain that we think is going to subsequently be removed, so it doesn't have any long-term consequences. The microelectrodes themselves each extend approximately a millimeter into the cortex, so we don't know for sure exactly which layer they are recording from, but we think that it's approximately layer 3 or layer 4 neurons in that part of the lateral temporal cortex.
Thanks, Karim. The next question I have here is for Jen. With your decoding approach, would someone be able to change the amount of force that they are applying to an object?
Yeah, thanks for the question. So yeah, we've actually looked at this a little bit because the way that the task is designed that I presented was the hand starts open, at least they visually see that, and then it closes and starts to apply the force. And we initially measured that transient response during that closing phase. So we weren't sure if that was related to the kinematic movement of the hand. But actually, even during an isometric grasp, when you're holding on to the object, if the user wants to increase or decrease that force, we see that they enter back into that same transient dimension. And so we can use a single decoder to either apply that initial force or change it as you go along. And so we're not really switching back and forth, but we're looking at the probability of whether you're in that transient state or not. And that tends to be high, again, if you want to change your force.
Great, thank you, Jen. And we are going to wrap up the question and answers with that last answer from Jen. And again, I want to thank all of our speakers. We really appreciate you taking the time to work through the pre-recording and joining us today for the live question and answer. It's wonderful that we can all do this virtually in this very sort of different time that we're in right now. And thank you to the audience for your participation and your attention, and I hope everyone stays well and enjoys the symposium. Thank you.